I'm Kendra Zuberbuehler, and I was lucky enough to get the opportunity to work with AAI, the American Association of Immunologists. I'm a high school science teacher at Middle Creek High School in Apex, North Carolina, and I teach chemistry. And I was offered the opportunity to have an internship at Duke University with Dr. Lee Reinhardt studying immunology. And part of this internship was to show how immunology can be used in my classroom. Now, many of you, or almost all of you, are biology teachers, and I'm chemistry, so how does something like immunology relate to my field? So what I looked at is the study of light and fluorescence in the instruments of immunologists that I can show my students in my chemistry class. So how do I connect immunology to chemistry? Well, I just look at the light. So our students have to learn the general equation of energy is equal to Planck's constant times frequency, and the speed of light is equal to wavelength times frequency. We talk about Bohr's model of the hydrogen atom and how that energy can be given off when electrons relax back down. So we have our model of our hydrogen atom, and then the rings of energy levels around that. Electrons absorb energy to increase in their energy levels, and then as they relax back down, they emit a photon of light at different wavelengths depending upon what wavelength of light and what energy transition that is. And so we demonstrate that usually by burning salts, and it's kind of the classic science experiment to show that. But it really is used in immunology and in biology with fluorescent staining of antibodies on cells. So the cells in this case are our lymph nodes. And lymph nodes can have fluorescent tags attached to the antibodies to be able to tell what kind of cell is inside of our lymph nodes. And so I used 3 different instruments at Duke, and I wanted to model to my students how they can see the chemistry being applied inside those instruments. So I'm going to show you, we did 2 labs, and I gave them some background information before we did those labs. So I'm going to show you just what we did as a class with my sophomores and juniors here in a chemistry classroom. So first they had to learn what is this immune response. And the way I did that is I showed them exactly what happens in the immune response. So in our immune system we have T cells and B cells inside our lymph node. And they first learned that B cells come in to contact with a virus, that B cell breaks apart the virus into pieces, and the pieces get presented on the outside of the B cell. Then a T cell comes in, and a T cell and a dendritic cell come together, and the dendritic cell engulfs the virus. It presents the virus outside of it. T cell comes into contact. T cell gets activated. Then my B cell, my activated T cell, come together. It activates my B cell, and now my B cell tells the rest of the body, hey, there's a virus. Something is wrong. And so it's this pathway that our body uses to develop an immune response, but we can't know this, and we didn't know this unless we were able to actually see and follow our T cells and B cells around. So immunologists have tagged B cells and T cells with these fluorescent fluorophores that emit different wavelengths of light when they're excited. One way that our students were able to see this is by looking at flow cytometry. So normal flow cytometer has a cell sample brought down into it. It passes through a laser. That laser excites the fluorophore on that cell, and that then light gets picked up by one of the photomultiplier tubes and sent to the computer. But we don't have that, as many of you probably also don't have, so we made a model, and I actually have the model here with me since I'm here in my classroom and not with you. And what it looks like is I have a cardboard box, a bottle of corn syrup, a lymph node, a Erlenmeyer flask with a test tube, and a light. So you can see here I have my box. It's just a normal box. I put in some paper and grid paper on the inside. Each student received a bag of lymph node cells. The lymph node cells are just glass beads that I got from Michael's in different colors, the blue being B cells and the red ones being T cells. Activated T cells had a green tag on them. Activated B cells had a yellow tag on them. So students were given their bag of lymph nodes and their flow cytometer. In their flow cytometer they had their test tube, and I diluted my corn syrup about 10% so that way the beads fell faster. And in the dark room they took their laser, which was their flashlight, they shined it against their test tube, and one by one they dropped their beads down in, and as it passed by, the light being pushed through is shown on the back, and you can kind of see the green showing up and then the red. And my students recorded the data of the different colors emitted by the cells that they dropped down. Afterwards, they counted up how many they had of each, and they determined the state of the immune response. So just regular T cells and B cells, no immune response. Activated T cells and B cells were having an immune response. So that was the first experiment that they did. Our second experiment looked at fluorescence microscopy. So fluorescence microscopy is where we take a lymph node, and they did a color by number. So I drew a lymph node, and then I went ahead and I numbered it, and they had to color in the different cells within the lymph node depending upon their instructions. Now their instructions were to color T cells one color, B cells another, activated T cells one, and activated B cells another. So these are my 3 different cells, and I can provide you with PDF versions of these if you'd like them. And they were told to color them. So first they colored the T cells yellow, the B cells black, because this is our B cell zone and this is our T cell zone. This is a healthy lymph node. There's no activated T cells or activated B cells. Then they went ahead and they colored by numbered their next 2 lymph nodes. And I had them use highlighters. That way the ones that were activated fluoresced. And so we actually looked at them underneath a black light, and I have my black light here. And when they shine their black light on their lymph nodes, the highlighted one fluoresced just like they would under a fluorescence microscope. So here just a couple of activated T cells, a couple of activated B cells. This is in the middle of the immune response, and then a lot of activated T cells, B cells, and germinal centers forming. It was a very high immune response. So they were able to see the different kind of immune responses, and then again be able to tell what kind of immune response was present in these cells. When we talked about how this would be really done in the lab, staining cells, we had wait times, and they were able to see what the process was like in a real immunology lab. My students really enjoyed what they did. A couple of them are now in contact with Duke, talking to them about opportunities that they could have. And so if you have any questions with this, feel free to email me. My email is here, and it'll get put with my curriculum as well. I just want to thank the American Association of Immunologists and their high school teacher internship program for funding me. This experience was great. I really enjoyed it. I'd like to thank Dr. Richard Lee Reinhardt and his lab, Tommy, Catherine, and Mark, for just bringing me in and showing me the ropes. And then my husband, Matt, he's also a PhD student in immunology at Duke, and he spent a lot of time explaining the different roles to me. So I'm sorry I can't be there with you. I would love to be there, but I will be here at school teaching my students tomorrow. Any questions, like I said, just let me know, and thanks for watching.